Hello students, in this video I am going to discuss with you the translation. Okay, translation, it's a part of central dogma which describe the process of synthesis of proteins from the RNAs. Okay, so there are three types of RNA we know, right? mRNA, rRNA and the tRNA. So messenger RNA, ribosomal RNA and the transfer RNA. All these three RNAs involved in the process of protein synthesis, that is translation. Now we are going to see the mechanism how it produced. Okay, first of all we need to see the ribosome. Ribosome is actually the site for protein synthesis. It is the factory for protein synthesis. Okay, ribosome. Now this ribosome, it has two structural units. So first of all let me draw the ribosome here. Okay, this is a two structural unit of ribosome. Okay, this is the large subunit, this is the small subunit. Okay, so let's label it. This is the large subunit and this is the small subunit. This is the size, you can say 60s, and this is the size of 40s. Okay, two structural unit of ribosome. Okay, and both of these involved in the process of translation. Okay, so now here there is a gap, you can see, when these two structure, okay, comes here in attachment. Okay, trying to come in attachment here, this is only happen when this process, the translation about to start. Okay, these two structures comes over each other like a cap, you can see. Okay, now here the mRNA, the messenger RNA, okay, is there, it get fixed in between the two structure of the ribosome. But before that we need to see, okay, there are some structural locations. Okay, let me first draw the mRNA here in between the two structural unit of ribosome. Okay, just one second. Okay, so now here this is the mRNA strand, and you can understand that mRNA, which is a strand having this sequence of nucleotide molecules, that is the nitrogenous bases, or here now we can use, technically you can use the term of codons. Okay, so let me show the codons or the sequences, whatever you say, there in the mRNA strand. Okay, now the important thing is that you're in this structural unit, large subunit of the ribosome, it has three sites. Okay, A site, P site and E site. Okay, this is the A site, this is the P site and this is the E site. Okay, so this is the location here. You can think of this area is as the E site. Okay, where okay, let me just make it a little more okay, so this part is the E site, this is the P site, this is the A site in the ribosome. Okay, now A means amino acyl tRNA site. Okay, A for amino acyl tRNA site. Okay, whereas P for peptidyl tRNA site. E is for exit. Okay, we will see the importance of these sites. See, first of all, this mRNA, if I give it a 5 prime, it is the 3 prime here. The mRNA needs to fix in the ribosome in such a way that its AUG codon should be placed at the P site. You have to keep in mind this, okay? The mRNA should be fixed inside the ribosome in such a way that its AUG codon should be there at the P site, okay? So AUG, okay, this is site here, and before it, after it also there are consecutive codons are there. Any codon you can write here. After it, AUG, and even before, but we have to keep in mind that the AUG as the initiator codon, as you know, the translation will start from here, it will move into that direction, not into this direction, okay? But the codons are obviously there one after another. Specifically, the AUG codon should be there at the P side. This is how it needs to be placed. The mRNA is placed actually inside the ribosome, okay? Now what happens is that the tRNA, okay, the tRNA which are there in the cytoplasm, it is there, and specific amino acids needs to be taken, okay, from the cytoplasm. Specific amino acids needs to be taken inside the ribosome so that it can undergo the process of protein synthesis. So one after another we will see the process. First point, if I discuss, the amino activation of the amino acid. So first thing is the amino acids and the tRNA both are there in the cytoplasm spreading, that is outside of the ribosome in the cytoplasm. Amino acids are there, even tRNAs are there, okay? This amino acid first of all needs to be activated. Activation of amino acid is very important, okay, at first. So how to activate the amino acids? With the help of enzyme, with the help of ATP mainly. So the amino acids, this amino acid I am writing here, needs to be binding with the ATP, okay? With the help of the enzyme we can name the enzyme as amino acid synthetase enzyme, okay? Amino acid synthetase enzyme, okay? And magnesium ion is also important. One thing is that remember, when the two subunit of ribosome get attached with each other, there is also contribution of the magnesium ion, okay? So magnesium ion here it is required, and the amino acid synthetase. This enzyme in the presence of magnesium ion helps